DNA, meaning the double helix, purines, pyrimidines, and all that good stuff. But it was a lady by the name of Rosalind Franklin. Look her up. Lady by the name of Rosalind Franklin did X-ray diffraction and discovered that it was kind of this doubled over appearance. Without her work, they would not have been able to do a thing. How much do you hear about her? You know, that's what I'm saying.
Is she the one that got a Nobel? Oh, no, she must not have got a Nobel.
Rosalind died.
Franklin, right?
Rosalind Franklin.
Yeah. Why don't we now? All right, Katie's in the lead. Term for the subunits that make up the DNA. Nucleotide bases or nitrogenous bases, but nucleotide is the proper term for those. All right.
So they're not bases, they're base pairs.
Correct. Correct.
It was just
Which, no, but that was like I said, that's why I said I didn't make them up, because that's a little misleading. But yeah, nucleotide's the better answer for sure. All right. What's the name of the sugar that makes up DNA? Deoxyribonucleic acid. Very good, very good. All right, so here we go. Which of these are purines? Why does it matter if something's a purine or a pyrimidine? It has to do with the way that they go together. So what Watson and Crick did was they realized that if you had two purines, it was going to be too wide for them to bond. If you had two pyrimidines, it would be too narrow and they would overlap. The only way for the strands to come together and make sense is to pair a purine with a pyrimidine. So A always pairs with
T.
T. C always pairs with G. G. And which two are purines?
A.
A and G. All right. Tell me the complementary strands.
Wait a minute, this is more time.
I'm hoping that if you missed this, it was because of you just missed time. But you know how to predict the new strand. And it's based on that principle of complementarity, which means what? They go together. They complement each other. Somebody came up with rules. Who was it? Chargaff's rules. So that means you always pair A with T, C with G. Good, good, good. All right, Crystal is in the lead. Whose observations? I think I may have just given this away. Oops. Shargoff came up with the principle of base pairing, A with T, C with G. Good. KT is ahead. What makes up a nucleotide?
How are you? I know. I got it last night. I didn't get any though. Unfortunately.
Yes, this one, all of the above. It's a pentose sugar, 5-carbon sugar, phosphate group, and a nitrogenous base. And that's where the distinction is, because the nucleotide includes the section of the ladder, and the nitrogenous base alone is the run. Does that make sense? Okay, and I'll show you that again when I draw the structure. All right, very good. How are base pairs of nucleotides held together? Yeah, they're hydrogen bonds. Why are hydrogen bonds significant here? What do you remember about them?
Because they're
They usually have to do with water, but not necessarily. If there's hydrogen involved, it's a bond that's not particularly strong. So it's a little bit more tenuous than, say, a covalent bond where you have things that are tightly held. And that's important because it allows for things like DNA to come apart for replication and so forth. So hydrogen bonds holding the ladder together. What is the name of the process that makes an exact copy of DNA? It is replication. Duplication, replication is a better word. Synthesis, technically, yes, you are synthesizing something, but when we're talking about DNA, it's more appropriate to say replicate. It's a replication. All right, very good. What's the complementary RNA strand?
Be the yellow. I think it'd be the yellow.
Really only have to do the first one. Okay, good. Most everybody got that one. What's the difference between DNA and RNA? RNA uses uracil in place of thymine. You know how you can remember that? Have I told you this already? Oh, yay. If I had to choose between DNA or RNA, I would choose RNA because it has U in it. Right? Yeah, so there is no thymine. Good. What is the term given to a 3-nucleotide sequence that codes for a protein? I see 2 of these things working. I was giving triplet for that one too, because technically, you see them referred to as triplet codons. Codon is more precise, but I think if you said triplet, that's equally correct. I would have gone either way with that one. Very good. All right. I love this question. This is one of my favorite test questions. 15% adenine. What percentage is cytosine, or is there enough information to figure it out? Math is power, y'all. How do we figure this out?
adenine doesn't pair with cytosine, so there's obviously 35% of both cytosine and guanine, and 15% of adenine and thymine.
Yeah, Everybody get what you got? Yeah, so if you've got 15% A, that's what it is, right? Add D. Okay, so if you got 15% A, you know that A is always equal to
Like, what is it?
T. So that means you got 15% T for a total of 30%. So that means that the remaining
I
two
totally forgot
had
about
better
that.
add
I'm
up to
sorry.
be 70%.
About the videos.
Which means each of them contributes 35%.
No, you're not bothering me. I keep forgetting.
I promise you'll have a question like that on your next test. It won't be admin, it won't be 15%, but it will be similar. Very good. Image below is an example of what? Yeah, and I told y'all not to worry about the chemicals. Let's just skip this one. It was a, and I specifically said, don't stress about the chemical formula. So again, these aren't my questions. That'll do it. All right, an RNA, the base thymine is paired with what? Y'all better not say something. Just say it. Yeah, this is misleading because it makes you think that you're developing a similar strand. That if you're making a strand of RNA, thymine is gonna get paired with adenine. That's a trick question. That's a trick question. See it?
Say that again.
The question is, it makes it look like you're synthesizing a strand and matching thymine with DNA, but you're not.
You're just copying.
You're making a complementary strand with the T in place. It is, don't worry about it. All right, who won? KT won. Is that you, Lawrence? No, that's you. I know, you're so surprised. No, I'm not. I'm not surprised. That's awesome. Yeah, we won't worry about that either. Okay, so, looks like you did your homework, which is outstanding. So that means that now we get to talk about genetic material and how it works. I'm gonna do most of this on the board, and then I'm gonna fill in the holes with the notes, because this is the way I like to do things. Y'all all saw your announcement about the next exam, when it is, what it covers, and all that good stuff, say yes. Okay, you should. It covers chapters, these 13, I'm dropping out of that group, and I'm gonna give you an activity or something to do for that chapter instead. And we're not gonna devote a ton of class time to it, because 13 is easy, and we need to spend our time talking about complicated stuff, yeah.
So will that assignment for 13 be great? Like, it'll be something.
Yeah, it'll be something you can do that's not punitive, but that covers that material.
So will it be on the exam?
I don't remember if I said so or not. I think most of it is. It's general stuff. And the activity that I give you, Sydney, will kind of show you, it's like disinfectants. What's the definition of disinfectant versus antiseptic and that sort of thing. So it's not. I don't remember what I said about it. It's in the announcement. Yeah. Okay, yes?
That's chapter 10.
This is 11. Okay, so let's take a look at these three kind of processes that we're gonna be dealing with. So, and I'm gonna make a point about this because every semester, students mix these things up. And even though they're covered in the same chapter, one sort of follows the other, but replication is a process all by itself. So the first part that we're gonna deal with is DNA replication. Which literally means what? You're making an exact copy of the DNA, and why in the world would you want to do that? Say again?
Grow.
Yeah, because every cell needs a complete copy for it to work, and this is the process by which you get that exact replicate for each new daughter cell. So replication, making a complete and hopefully exact copy of the DNA. Following replication, when does replication happen? Who remembers? Talking cell cycle. Thank you. It's the S phase of interphase.
Yes.
S phase of interphase, DNA replicates first, and only after it's been proofread does the cell reach the mitosis checkpoint, which allows it to proceed through the rest of the division. So replication is a separate entity. It's over here. It's all by itself. It has nothing to do with the following processes. You got it? Alone. So the next thing we're gonna look at is transcription, which by definition is what? Yeah, transcription is the process by which we take our DNA blueprint. Remember the cell analogy that I gave y'all? NAP1, that was the North Pole. So the North Pole is the nucleus, the DNA is Santa Claus, and the DNA has the instructions for building the toys. In this case, the toys are what? Proteins. Proteins, you're right, you're ultimately right, yeah, they're proteins, right? But before that can happen, the information in the DNA has to get out of the nucleus. Well, we can't send a strand of DNA out into the cell, right? But we can send a copy of the instructions. In other words, your architect draws up the blueprint and sends that to the builder, right? So DNA gets copied or transcribed into mRNA, and the M stands for messenger, because it's literally carrying the instructions out into the cell. So then from there, we have translation, which is mRNA being converted into what? It's tRNA? No, the rRNA and the tRNA play a role in this process, but the mRNA is the directions, and what comes from that is literally the amino acid sequence that is used to build proteins.
So your peptide.
Yeah, your peptides. So the DNA is Santa Claus. The instructions in the form of mRNA travel outside the nucleus to the ribosomes or to the endoplasmic reticulum, what have you, which is the workshop, and you put those amino acids in the proper order. Okay? Well, this notion of going from DNA to RNA to protein is what we refer to as the central dogma of all biology. What's a dogma? What if I told you that Baptists tend to be dogmatic? Like a rule? Yeah, it's like a rule. It's like a belief. It's something that you hold fast to come hell or high water, no matter what. This is the central dogma of all biology, which means that everything in biology, everything, is discussed with this in mind. Wait, what? Why is this such a thing? Why is this so almighty important? It doesn't look like that big of a deal.
Because everything has DNA.
Everything does have
That's
DNA.
how everything is able to grow and reproduce.
True. But think about what that DNA is ultimately used to do. It builds everything. Be more specific. What is it ultimately being used to do? It's protein synthesis. It's ultimately the instructions for building the proteins that do what? What don't they do? Right? All the chemical signals, all the antibodies, all the cell receptors, all the structures, all the messaging, and all the enzymes, all the chemical regulation is carried out by those things. So it makes sense that this is a big damn deal, because if you don't understand this process, how can you understand anything about how living things work? Not only that, but DNA is how this information gets passed from one generation to another. So it's heritable information. So that's why we spend so much time on it. Does this have anything to do with this? No. I want you to think about those as totally separate things. There's always a test question where it's, which of the following doesn't hinge on the others and what have you. Replication is a separate process. It's lumped in with the genetics chapter because it involves DNA. repair and all that fixing and what have you, but it is a separate entity from transcription and translation. The other reason they're put together is because if you understand replication, transcription and translation are much easier. Alright? Okay, so let's see how it works and what some of the implications for it are. Just as with any chemical reaction, we have players that help mediate, and who are they? What class of chemical make the reactions go? Enzymes, yeah, so we're gonna see a whole series of enzymes at play here. The first thing I'm gonna do is I'm gonna draw some DNA, and you should too. I'm gonna make this the 5' end, and this is the 3' end. Actually, no, I'm not. I'm gonna make this 3'. Sorry. I do this every time. What does the 3 and the 5 refer to? Did y'all get into primes in your, okay, you know what the sugar molecules look like. They're kind of hexagonal, and at each corner, there's a carbon, which indicates a sugar, right? Let's see if I can find one. And not. Okay, apparently not. All right, anyway, but the 6 carbon sugars have to be oriented a certain way for the strands to go together. The position, so you're looking at a sugar molecule that's kind of like that. It's understood that at each corner there are carbons, even if they're not written in, those are carbons. The position of the carbons is what gives the 5 and the 3. In other words, how this loop is oriented. Does that make sense to you? It may not make a ton of sense, especially if you're Jamie and detail oriented and you have to know every little thing. And that's okay, but suffice it to say that if they're not lined up with a 5 here and a 3 on this complementary strand, then the strands are not gonna go together the way they're supposed to. Okay, like they won't meet in the middle. All right? So we've got a 3' and a 5', and that's what it refers to. 3 always goes with 5 and vice versa. What is the term that describes how, if you have one strand of DNA, its complement strand is a mirror image that's also flipped? Anti-parallel. And the anti-parallel works because of where those sugars are, where the carbons on those sugars are. So if you had another 5' over here, it wouldn't bond together. Okay? So far so good? Alright, so when we copy the DNA, we don't just make a whole brand new strand. We start with a copy of the parent and we build onto half of that. Think about Legos. Who has kids that play with Legos? Have y'all ever had to stay up on Christmas Eve night and build a Lego structure? Or Christmas Day, assist with building a Lego structure? Yeah, it's fun. It's fun. So let's say you're gonna build Hogwarts Castle. Would you rather start from scratch with just a picture? Or would it be nice to have half of it already built and all you have to do is copy it? Yeah, half already built. They don't come that way, by the way. But half already built is the best. And so that's what we do with DNA. We split the double helix and we build onto the existing strands. What is the term for that type of replication? It's semi-conservative. Semi-conservative, meaning you're conserving part of the parent strand and only synthesizing the new one as a half. Okay, so far so good? All right, so here are, go ahead and draw in some, what were these called? Good, nucleotides. Draw in a few of them here, I'm just making these up. So if this is a C, this has to be a G. If this is a G, this has to be a C. C, if that's a T, that has to be a, why not a U? Yeah, because it's DNA. We can use adenine in DNA. All right, so I just made up some base pairs. Now these are the enzymes that we're going to be working with. All right, so our players, we've got gyrase. We've got helicase. We've got polymerase. We have topoisomerase. We have single-stranded binding proteins. And I think that's all.
What's under polymerase?
Topoisomerase.
Huh?
Topoisomerase. We have ligase too. I think that's all. Pretty good little set of players there, right? All right.
I can't read it, sorry.
Polymerase, topoisomerase, single-stranded binding protein, and the ligase. Write them down, draw it with me. Taking a picture's not gonna help. Yes? What? We are setting the stage for DNA replication, and we're introducing the structures and the players before we figure out who does what.
So this was the left side of that column we had. Where you had replication.
Yeah, this is all replication. Yeah, that's actually a good question. Does everybody understand where we are and what we're doing?
Yeah.
We're setting the stage for replication. This is the setup with our DNA, how it goes together and how it looks. These are the enzyme players. Yes,
What's the fourth one say now?
topoisomerase.
Topo.
It's spelled exactly like it sounds. Topoisomerase.
Thanks.
Sure. You can't read that?
Uh-uh.
Topoisomerase. All right. So before we can start replicating, we have to get our double helix apart. We have to be able to pull it apart and keep it stable. Now, keep in mind that on downstream, it's the double helix again. It only opens up the part that you're replicating at that time. It's sometimes referred to as a replication bubble or a replication fork, if you're looking at this place right here. So, the first thing that has to happen, the DNA has to untwist, right? It's a chemically bonded thing. It is most stable when it's in that double helix structure. So topoisomerase, gyrase, first of all, is what untwists it. So we'll put gyrase up here. Gyrase untwists. But the tendency is for it to twist back. So topoisomerase, I'm just gonna call it TI, finds right here where the replication is gonna go to keep it unwound or untwisted and stable. Make sense? Kinda is what it is. Single-stranded binding proteins also attach.
So basically the topo keeps it apart.
Yeah, topo keeps it from twisting back. The single-stranded binding proteins keep it apart. Okay? It just keeps it from twisting back. Because it's a twisted ladder is what it really looks like. So it has to be untwisted and straight for replication to work. Topoisomerase keeps it untwisted and flat. The single-stranded binding proteins keep these separated because they want to go back together.
So like right now.
Yeah, kind of. Actually, what came into my mind was a speculum, which is
Yeah, we
just a
should
horrible,
say.
horrible thing. But it's, you got it now.
If we came to my house, we would, for
See how
sure.
this looks like a ladder that's been twisted?
Should have a
DNA
huge stomach.
gyrase untwisted. The topoisomerase keeps it apart and keeps it from retwisting. Single-stranded binding proteins keep it apart.
That makes more sense seeing.
Yeah, a better picture.
And the replication
Best friend.
is
Yeah, she lives
Just
with
the
me.
place right here.
Yeah.
It's a fork in the road.
Yeah.
That's the gyrase, the replication.
She's taking
No, the
pictures,
gyrase
she's
just
taking
unwinds it at
one.
that place.
And it being the phosphate.
Sarah's taking it out of
What?
Highlands.
Yes,
Georgia
yes,
Tech.
the double
She
helix
lives there.
itself. Alright, so who untwists the gyrase? Who keeps it from retwisting? Topoisomerase. Who holds it apart? Single-stranded binding protein. Alright, we got single-stranded binding protein. Alright, so, at the same time, yes, sequential. You have gyrase, topoisomerase, binding proteins, everything kind of converges on the DNA and starts this process. Does that make sense? So it's all happening at the same time to keep it stable. Uh-huh, yeah,
The gyrase untwists it and unzips it?
no,
It just untwists
we haven't gotten to the unzipping.
it. Okay, it just kind of looks like where,
It
where
does, I know,
it is.
it's all
I
happening
just
at
wanna make
once.
sure I got you.
Yeah, gyrase untwists, helicase unzips.
Okay.
Which brings me to my most favorite biology line of all time. Are you ready for this? Are you sure? If I were an enzyme, I would be DNA helicase so I could unzip your genes. That's how we play. No, my handler did not use that line. Somebody's gonna ask, I know. Alright, so helicase unzips, gyrase untwists, topoisomerase keeps it untwisted, single-stranded binding proteins keep it apart. So we checked off topoisomerase, these, this is what the hell we're almost done. So far, so good?
It's
It's all happening all at the
It
same
keeps it
time.
from twisting. Single-stranded binding proteins keep it apart, and topoisomerase keeps it untwisted.
What's the difference in untwisting
Untwisting is this, unzipping is pulling it apart.
and? Okay, so once it's untwisted, then it gets unzipped.
Yeah, it's all kind of at the same time.
Right.
I mean, you got all this stuff, yeah, going on. Yep.
So there isn't a step that we need to sort out, like we don't need to put in this gyrase, topoisomerase.
No, you just need to know who's doing what. So these are the enzymes that are setting the stage for semi-conservative replication. Yes?
So the single-stranded binding protein keeps it from, going back to.
After it's unzipped.
After it's unzipped.
After it's unzipped, yes. Yes.
But the topoisomerase.
So rules all over the place,
Keeps
but I
it
get
from
the calls
retwisting.
at smoke shops.
So group your
Just different
helicase
flavors.
and single-stranded binding proteins together. Helicase unzips it, the single-stranded binding proteins keep it apart. Gyrase goes with topoisomerase. Gyrase untwists, topoisomerase keeps it from twisting back. Okay? And all this happens at once. We haven't even gotten to the good part yet. Okay. So now, just like with any other molecule, you have free nucleotides floating around, just like you did the adenosine diphosphate to make the ATP. So if you start wondering where those are coming from, they're already there. You consume nucleotides and everything you eat, bacteria are no different. So, polymerase, which is easy to remember because a polymer is a sequence of repeated parts, right? Polymerase is who's going to be adding those base pairs to the new strand, making the polymer. Okay? Now there are distinctions between polymerases. There's a polymerase 1, there's a polymerase 2, there's a polymerase 3, and they all do different freaky things. For us, it's polymerase. We're not gonna make the distinction between the three. If it's adding base pairs, it is polymerase, okay? There's a catch though. Polymerase can only add to a particular shape of molecule. Remember when we were talking about molecular shapes with our 5 and our 3? Yeah, what does the 5 and the 3 refer to? The position of the carbons in the sugar, okay? Polymerase can only add to the 3' end. So that's what forces the direction to be specific when you're talking about DNA. So what I'm getting at is, here, we've got a 3' end right here, correct? Say yes.
Yes.
Okay, so what's our new end gonna be technically?
5.
Yeah, so the 5' is gonna be the complement to that, which means if you can envision this being that hexagonal sugar, in fact, I'll write this. Oops, that was terrible. There we go. All right, so if this is the 5' side, this side must be the what? The 3. Okay, does everybody understand that? It's just a given. If you have a 5' carbon on one side and a 3' on the other. If this is the 5 prime, it has to be 3 over here.
On the other side of that shape, like directly across from the top,
Yeah, well, yeah, this is the sugar on, the carbon on the other side.
is the three.
Yeah, is the 3 prime. Yeah. Yeah,
So
so
another one, what they get, so it'll be 5 on that side. Right?
it has to do with where the base pairs are added. This is part of your sugar phosphate backbone.
Okay.
So that means that polymerase can add to this end because this is the 3 prime end. Since you're adding to the 3 prime, you can just add it all the way along.
Explain this prime thing to me, because I'm just not picking up what you're saying.
Everywhere there's a corner on this hexagon, there is a carbon. And you number those 1, 2, 3, 4, 5, depending upon the shape of the molecule. So 5 prime refers to a carbon being in that 5 position. 3 prime is a carbon in the 3 position.
Okay.
So it has nothing to do with other than where they're positioned on the sugar molecule.
It is where they're positioned on the sugar.
Yeah, that's what I'm saying,
That's
that's
all
what
it
it
is.
is.
Yeah, that's all it means.
Yeah.
That's all it means.
That's what I'm saying.
But the reason it's important has to do with how these strands match up.
Right.
Because you can't have you could not have this sugar flipped and then it match up and fit. It has to be reversed and anti-parallel. And that anti-parallel is because of where those sugars are. Does that make sense? Okay. Now, the other reason that it's important is because polymerase can only add to this side of the sugar. It can only add to the 3 prime end. Well, luckily here, we have the 3 prime end exposed, right? So that means that polymerase can come along and start laying down base pairs with no problem at all. And then it continues in that direction. It's smooth. It's continuous. This is the leading strand. Leading over here. It's the leading strand because it's continuous. There is no interruption. Polymerase just starts laying down those base pairs. That's all. That's how it works. Think about. Y'all know what poppet beads look like? You know those little beads that little kids play with that snap together, and they look like this. You got one end that's got a hole, and the other end kind of has a sticky outy part. Y'all know what I'm talking about? And the beads have to fit together a certain way. That's how the 3 and 5 prime work. If you have this bead turned that way, there's only one way you can add another one to it, right? That's how 3 and 5 work. If it's flipped the other way, and all you have are beads with the boy part on the left, you can't add to it. Well, that's what they look like. I mean, y'all know what I'm talking about. Think of the 3 and 5 that way. So continuous synthesis. You're just tacking, that's the leading strand. It goes in the direction of the replication fork and continues until you get to the end, right? What is a primer? You paint, what does the primer do?
Base.
Primer is the base coat.